Hi, I'm Thorin Tabor. I'm a computer scientist and software engineer by training, but these days I work with bioinformaticians and various data science tools applied to biology. I'm part of the JupyterLab, which is currently a collaboration between the University of California, San Diego, and the Broad Institute of MIT and Harvard. Today, I'm going to be talking about GenePattern Notebook, which is a suite of Jupyter extensions and associated libraries that we have developed to make working with JupyterLab easier, more reproducible, and more collaborative. To begin with, I like to talk about what I call the handoff problem. This is something that many, dare I say most, Jupyter users have ran into. So, by way of example, imagine you have a notebook that you've just finished creating. In this case, it's a notebook that uses the SVM machine learning method to predict and identify subtypes of leukemia. You've spent a long time working on this, but your code is finally ready. You've got your discoveries or insights, and you're ready to share your notebook with a collaborator. It could be another researcher, it could be your boss, it could be an editor for a paper you want to submit to, or whomever. So, you save the notebook and send it along, confident in your method. Unfortunately, they can't get the notebook to run. Or maybe after a lot of struggling they do, but they get different results. I mean, it worked on your machine, but then you had NumPy installed. Or maybe you're in Python 3.8 and they're still, for some reason, using Python 2.7. Or maybe you've got mutually incompatible versions of Plotly installed. I've personally run into all of these. Now, to get around this issue, there are some best practices that have become common. For example, you could check your notebook into a Git repository and give it a requirements.txt file, listing all of the notebook's dependencies and dependent versions. You could write a README file that explains the version of Python to use or what conda channels you need to install packages from. You could even include all of this as instructions right at the top of the notebook. But that brings us to the next part of the handoff problem, which is solutions like requirements.txt files or conda environments actually require a fair amount of technical skill. I mean, maybe not for the seasoned programmer, but not everyone's a programmer. Even in research organizations, there are going to be bench scientists or product owners or management who may not have the same prowess on the command line. And often it would be to your benefit for them to be able to reproduce your method, either because they have the best domain knowledge, or they're reviewing a paper, or so that your work doesn't present a bottleneck, or they're writing your paycheck. So, to summarize, the handoff problem exists because notebooks don't fully encapsulate the environment in which they are executed. There are outside dependencies and assumptions that can get baked into the code. And handoff can also be difficult because not everyone has the same technical skills. The more we can ease these burdens, the more reproducible your notebook will be. And I want to point out that in the world of reproducible science, this is especially important. So, let me tell you a little bit about who we are. As I mentioned earlier, I work in the JupyterLab, and one of our main foci is easing the burdens of reproducible science. To that end, we produce an open source piece of software called GenePattern. Simply put, GenePattern is a platform for taking different analysis or machine learning methods, encapsulating their environments, and putting them behind a user-friendly interface so that users of all levels of technical sophistication can run computational workflows without needing to worry about dependencies or the command line. We've been at this for a while. GenePattern predates Jupyter's web interface. And a few years ago, when Jupyter began to get big, we started using it. And it was pretty amazing. In particular, I was impressed with its ability to interleave text, images, and other narrative elements with the computational steps of a workflow. Because of that, we thought it was a good fit for our mission of reproducible science. We began to integrate our GenePattern tools into the Jupyter ecosystem, and thus GenePattern Notebook was born. We've come a long way since then. Originally, we were writing tools for Jupyter Notebook, and we still support it, but recently we're focused on JupyterLab. We produce a variety of tools that are aimed at improving the Jupyter experience, thus reducing its technical barriers. We use these tools to work with cancer data, looking into the gene expression of different tumor samples, but I want to emphasize that the majority of the tools I'm going to show you today are domain agnostic. That is, they should be equally applicable regardless of your field. GenePattern Notebook consists of a suite of JupyterLab extensions and associated libraries that extend the familiar Jupyter interface. The goal is to make computational analyses easily accessible to users at all levels of technical sophistication. On the back end, GenePattern encapsulates the environment of each step in a programmatic workflow in a GenePattern module. Each module captures the environment in a Docker or Singularity container, executes the analysis inside that container, and makes any result files accessible to the user. Depending on what options you employ, analyses can be run on the same machine, or on a remote machine, allowing for different hardware such as large amounts of RAM, CPU, or an entire compute cluster. On the front end, GenePattern Notebook employs a user-friendly interactive widgets. These provide an interface to programmatic functions or calls to external services. Or they might represent the results of analyses, displaying tables, interactive visualizations, or linking to external files. We also have a suite of Jupyter Hub extensions that help enable collaboration in a multi-user environment by providing notebook annotation, publishing, and sharing capabilities. One of the underlying goals is to encapsulate a complete research narrative from its conception to its dissemination. This way you can leverage the best of what Jupyter and GenePattern have to offer, interleaving text, graphics, interactive widgets, visualizations, or other analytic aspects. Now, before I get too far into the details, however, I first want to take a look at a real-world example of these tools in action. Here you see a screenshot of a fairly typical Jupyter notebook. What it's doing is loading gene expression data from various tumor samples and then performing support vector machine (SVM) analysis to divide those samples into different classes. These classes should align the different tumor types. This is first performed in a set of training data where SVM attempts to find an optimal hyperplane for dividing the samples. It's then given a new set of samples of unknown tumor type. The hope is that it will then be able to predict the type of these samples based on the hyperplane chosen with the training data. Now, if you didn't follow what I just said, that's fine. I'm not going to quiz you later. But I do want to note that SVM is pretty common in machine learning these days, and this particular notebook uses the implementation in the scikit-learn package. Anyway, this example works, but it's also about 70 lines of code. And if we're going to reuse this notebook, that code needs to be maintained. Furthermore, if I'm saving this notebook and sharing it with my less programming-inclined colleagues, it would be a challenge to explain to them how to modify it and use it with their own data or to set different SVM parameters. Here is another notebook. It's also doing SVM analysis, only it's using one of GenePattern's interactive widgets. Analytically, these two notebooks are identical. That is, they're both performing SVM analysis on the same training data, using the same test data, and will reproducibly yield the same results. In this notebook, however, the SVM analysis is being rendered as an interactive widget. Documentation is available through a graphical menu. And if I share this with my less technically inclined colleagues, it's a lot easier to explain. Just click the upload button, select the data you want to use, and then click run. Even as a programmer, I like to sometimes use these widgets. If nothing else, they help to reduce stupid, albeit time-consuming mistakes I might make in the code. All of this makes for faster and more accessible research. I like to say it's the quicker picker-upper. Now, one more thing before we take the deep dive into the specifics. I want to remind you that everything you see here is open source. Everything we've done in the last few years has been released under the BSD 3-clause license, although we still have a few older projects kicking around under other open source initiative approved licenses. So I keep mentioning that GenePattern Notebook is a suite of Jupyter extensions and libraries, and here is the specific breakdown. These include MB Tools, which is our interactive widgets framework, Jupyter WYSIWYG, which is a rich text editor for markdown cells, Notebook Repository, which contains Jupyter Hub sharing and collaboration functionality, and finally the titular GenePattern Notebook package, which includes all of our bioinformatics and machine learning offerings. And I'm going to go through each of these packages in more or less the order you see here. So to begin, let's take a deeper look at the NB Tools package and extension. This package allows a notebook developer to render Python functions as interactive widgets, both the input to a function and the output produced by that function. It also allows users to string together these functions so that they form a complete interactive analytic pipeline. NBTools is built on top of the IPyWidgets library, which is the core widgets library developed by the Jupyter team. It also includes a user interface where a non-programming user can add new analytic steps from a graphical menu. In my opinion, out of all of the features I just named, the ability to render functions as interactive widgets is the real star of the show, and we call this the UI builder. It quite literally allows you to take any arbitrary Python function and to render an interface to that function as an interactive web form. Then a user can just fill out the form and click the Run button to execute the function. This means that if you're a programmer, you can write your own code, then, with a single line, use NBTools to render a form-based interface to your method in a manner accessible to users of all levels of technical skill. And I want to note that it is fairly smart about how it does all this. It will infer expected parameter types from default values. It will display a description based off the function's docstring, and it will use parameter annotations to provide descriptive text. Additionally, you can override any of these defaults by providing a bit of metadata, and the user filling out the form can either use string literals or reference variables by name. The UI Builder even has the ability to upload and load files from disk by dragging and dropping the file to an input field. Now, you may be wondering what the code to produce the widget you just saw looks like. Well, it looks something like you see on the screen right here. In this case, we have a small Python function that wraps scikit-learn's k-means clustering object and fits it to a set of data. We've imported NBTools, and to this function we've added a single line of code, which is the decorator you see highlighted in bold. To display this function in a Jupyter cell, you simply need to leave the function as the cell's return value. The widget will then automatically be rendered. Alternatively, you can use the IPython kernel's built-in display method. In addition to turning Python functions into interactive widgets, NBTools also supports the ability to display text output, links to result files, or even display visualizations. We bundle this functionality together in the UI output widget. These widgets help maintain a consistent feel and help call out the important parts of a cell's results. This signals to users where their attention needs to be focused. These cells are also integrated with UI Builder widgets, and each output file has a menu where if you click it, the file can be sent to downstream steps in your workflow. To create one of these widgets, just return a UI output object in a Jupyter cell, passing in as a parameter any of the output values you want to display. Finally, NBTools provides an easy user interface for adding new interactive widgets, analyses, or other content to your notebook document. This interface allows a user to browse or search through the available analyses and adds the appropriate cell with a click of a button. We call this interface the Notebook Tool Manager. It has its own simple API, and in fact, UI Builder widgets are automatically registered with this manager by default so that their analyses show up in the list. You can also use this API to build up libraries of your own analytic tools and have them loaded into the interface. Just mark your functions with the annotator, and when you import them, they'll automatically show up in the list. And I want to take a moment here to note that we use NBTools internally in all of our analytic GenePattern Notebook offerings. So, even if you aren't somebody who's working in the realm of bioinformatics, the tools I'm going to talk about later can still serve as useful implementation examples of how to use the NBTools framework. Moving on to the next package, and along a similar vein of making things more accessible, we've developed an extension that provides rich text, what you see is what you get, WYSIWYG style editing for Markdown cells. This means that if you're working with someone who doesn't know Markdown, or if you just want to see a more graphic representation of the cell you're currently editing, you have that option available. It's presented in a familiar word processing style interface. Now, you can see from the screenshot that this is the one package we offer that hasn't yet been ported to JupyterLab. It's still using Jupyter Notebook, but JupyterLab support is coming in the very near future. I'm also excited to say that this particular extension has been picked up by SUSE Linux and has been made available through their YAST package manager. The next package I want to talk about is a bit different from the rest. Rather than being an extension for Jupyter itself, It's instead an extension for the Jupyter Hub multi-user server. We call this functionality the Notebook repository. It's essentially a web service that runs alongside Jupyter Hub and integrates with Jupyter Hub's Services API. This service provides notebook sharing and publishing capabilities to users of the Jupyter Hub instance. This is paired with a client-side extension that calls the service, allowing a user to browse the available public notebooks, to retrieve and run a copy of one of those notebooks, or to share one of their own notebooks with others. This makes Jupyter Hub easy to use in academic collaborations or any sort of group environment. And that brings us back to the self-titled GenePattern Notebook package. Now, I told you earlier that near the end of the talk I'd make a pitch for our biology and bioinformatics offerings, and this is it. But even if you're not in that space, you may be interested in some of the machine learning functionality that we provide with it. Anyway, what is GenePattern Notebook? The short answer is that it is the Jupyter gateway to the greater GenePattern ecosystem. GenePattern is a platform for reproducible bioinformatics research. It's a long-established project. The first release was back in 2004, and it's been going strong ever since. That puts it on similar footing to IPython, the parent to Project Jupyter. GenePattern is open source under a BSD-style license. Its repositories have a collection of some 250-ish analytic modules, including some of the most popular methods in genetics, genomics, transcriptomics, and proteomics, as well as general machine learning methods. Our public servers have some 75,000 registered users and usually run in the ballpark of 4,000 analyses each week. You can also download the code and run your own private server, as many organizations do. Finally, I want to note that it's an open platform. There is a repository of community-contributed methods that cover a wide range of bioinformatics techniques, including CRISPR analysis, bisulfite sequencing, flow cytometry, and RNAi screens. And by repping your method in the GenePattern module, it is automatically available in Jupyter through a user-friendly interactive widget. These analytic modules can be either ran on the local command line or packaged in a Docker or Singularity container. This means that GenePattern can execute analytic tools regardless of whether they are written in Python, R, Julia, C, or even Perl if you're that perverse. Our wrapper provides a standard interface to the analysis so that it can be called from a variety of programmatic environments. Behind the scenes, our Jupyter interface uses the GenePattern Python library, but we also have client libraries for R, Java, and MATLAB, as well as a web service that makes executing analytic modules possible through a RESTful API. For example, you can use these tools to run an analysis in GenePattern, retrieve the results, and then use that data with popular Python libraries such as scikit-learn, Plotly, Matplotlib, or Pandas. In fact, the code you see here is an example of running a job, retrieving its results, and loading them into a Pandas data frame. This also means that you are not limited by the analyses available through GenePattern. Using the provided analyses and coding your own programmatic analyses can be integrated rather seamlessly. And in fact, we provide data tools in our Python library for many common bioinformatics file types. These will recognize the type of data being loaded and automatically import it, including row and column headers, into a Pandas data frame. This is available both for files output by GenePattern analyses and for any other file the user possesses, so long as it is in a supported format. Once the file has been imported into a data frame, it can be viewed, filtered, or used in conjunction with numerous other Python libraries. Alright, so this all sounds good. Where do you go to use this functionality? If you just want to try it out to see how you like it, or if you work in the domain of biology, you can see our tools in action on the GenePattern Notebook workspace. No installation required. This is a free and public Jupyter Hub instance that we host for biologic and bioinformatics research. It comes with all of the GenePattern Notebook extensions, as well as many open source bioinformatics and machine learning libraries. And we host a collection of public notebooks demonstrating common methods such as single-cell RNA-seq clustering. It's available to the public at notebook.genepattern.org. To install the GenePattern Notebook extensions locally, they've been made available through the pip and conda package managers. If you install the GenePattern-Notebook package, you'll get the entire suite. Alternatively, you could opt to only install the NBTools or Jupyter WYSIWYG packages. We also have a Docker image with the whole stack set up and available, and that's up on Docker Hub. This container has been built for use either as a single-user Jupyter instance or in a multi-user environment in conjunction with Jupyter Hub and Docker Spawner. Finally, I want to reiterate that this is all open source. We have several GitHub repositories that are publicly available, and anyone is welcome to check them out. We love pull requests, I mean, who doesn't? Documentation is available right in GitHub and linked from each repository's README. So, to close, I want to take a look back at the handoff problem, at the larger Jupyter ecosystem, and at where things are headed. Now, if you remember back to the beginning of the talk, the handoff problem had two parts. That is, A, handoff is difficult because a notebook doesn't fully encapsulate the environment in which it is executed. And B, handoff can be difficult because of varying levels of technical skill between users. GenePattern Notebook eases these burdens and helps solve the handoff problem by A, encapsulating the environment used by each step in a notebook's workflow, and B, reducing the technical skill required to run and reproduce your analysis through the use of user-friendly interactive widgets. This makes your work more complete, more reproducible, and makes it more accessible to collaborators or other interested parties, regardless of whether they are project leads, bench scientists, clients, or reviewers. Now, stepping back to look at the greater Jupyter ecosystem, there are some great tools and a lot of pretty cool integrations. In particular, we know that real-time collaboration is in the works, and we're keeping an eye on that as it progresses. We're also continuing to port existing extensions to Lab, and look forward to many JupyterLab improvements in the near future. Finally, we've been experimenting with what we call our notebook project functionality, which is the ability to encapsulate individual notebooks and their dependencies in Docker containers, so that when you launch one, each notebook has its own environment. We think this will really help with reproducibility, and it will be released in the not-too-distant future. We're committed to making sure that GenePattern Notebook plays well with others and contributes to the greater Jupyter community. So, before I finish, let me just give a few acknowledgements to people who've contributed to GenePattern and to GenePattern Notebook. You can see the team on the left. Jill Mezarov is our PI. And on the right, you can see that we've gotten funding from these wonderful organizations. I would also like to put up a few resources from the net, which you can visit if you want more information. These include the GenePattern Notebook Workspace, our website, our GitHub organization, Docker Hub, etc. Finally, I want to thank everyone for listening to the talk. So with all that being said, enjoy the convention.